My first experience with addiction was when my father chose alcohol over his own children. When I returned to the reservation, I saw this heartbreak occur over and over. For myself and for my daughter, I choose to end the cycle. In order to do so, I needed to understand where it came from and why it existed in the first place. Prior to European colonization, alcohol was not a prominent substance in Native American societies. From Europe, Europeans brought with them distilled spirits and wine, which soon became valuable trade items. The introduction of alcohol plays a role in the misuse seen in Native communities today. Alcohol misuse contributes to higher rates of alcohol-related illnesses, such as cirrhosis, liver disease. Child neglect and domestic abuse are more likely to occur in households where a parent regularly consumes alcohol. Alcoholism coexists with depression, anxiety, and suicide. 11.6% of Native Americans reported an alcohol use disorder in 2023. 1 in 4 Native Americans reported binge drinking in the past month. Binge drinking, or alcohol abuse, is defined as consuming too much too often. Alcoholism, or alcohol dependency, is the inability to stop drinking despite consequences. Although historic influence is just one piece of the puzzle, we know that alcoholism is a complex genetic disease impacted by socioeconomic, mental health, genetic, and environmental factors. There have been adoption studies that show a person with alcoholic biological parents are more likely to be alcoholics themselves, even if they are raised by non-alcoholic families. These studies show individuals have genetic predispositions to alcoholism. But exactly how do genetics contribute to it? So let's think about the path alcohol takes in the body. It enters through the mouth, goes to your throat, into your stomach, gets absorbed into the bloodstream, and then it makes its way to the liver. In the liver is where the ethanol metabolic pathway begins. The ethanol metabolic pathway is a series of enzymatic reactions. The first reaction is ethanol, or alcohol, that gets converted into acetaldehyde. Acetaldehyde is a toxic substance, and we don't want it in this form in your body for too long. So it gets converted once more from acetaldehyde to acetate. Acetate is similar to acetic acid, which is vinegar. And then it gets converted from acetate to acetyl-CoA. Acetyl-CoA then goes into the Krebs cycle, and then what's left is used for cellular metabolism or energy. To summarize, we begin with alcohol. It gets converted into our toxic substance, acetaldehyde, and then it gets turned into what we can think of as vinegar, and then what's left is used for energy. For my research, I wanted to focus on the first conversion, ethanol into acetaldehyde, alcohol into an even more toxic substance. The enzyme that catalyzes this reaction is called alcohol dehydrogenase, or ADH for short. Simply put, enzymes are a result of DNA sequences. Changes in DNA sequences can lead to mutations in enzymes. There are 7 different ADHs in the human body, but I wanted to bring our attention to these 2 specifically. There's a variation in alcohol dehydrogenase 1B, and it's called ADH1B2. And this produces a faster conversion from alcohol to acetaldehyde. The overaccumulation of our toxic substance leads to symptoms like nausea, dizziness, flushing, headaches. Population, this is characteristic of Asian populations, and the people who possess this variant have a lower rate of alcoholism due to the symptoms that it produces. ADH4, on the other hand, is more active at higher concentrations of alcohol. When people binge drink, they often consume higher concentrations of alcohol. Native Americans have a high percentage of binge drinkers, and that is why we decided to study alcohol dehydrogenase 4, because of its link to binge drinking and to Native American population. Ideally, we wanted to find a documented variant of ADH4 in Native American populations. So we went to the Genome Aggregation Database. This website houses a collection of genomes from unrelated individuals of diverse ancestries. Yet Native Americans and other minorities are categorized under remaining. This inadequate amount of data is just an example of the marginalization that Native Americans and other minorities face in scientific research. If the data doesn't include you, solutions may not pertain to you. Because of the lack of data, we had to choose a different set of criteria. And in the database, we found ADH4 R375Q. R just means arginine. 375 indicates a location, and Q means glutamine. So basically this means that where there was an arginine in the normal ADH4, there is a glutamine in this mutant. Referring back to ADH1B2, there was an arginine that was changed to a histidine. And because of the similarities, we chose to study R375Q. So for our alcohol dehydrogenase 4 wild type, the conversion to alcohol to acetaldehyde is normal. For ADH1B2, the conversion to alcohol to acetaldehyde is accelerated, and that's why we get the buildup. ADH4 R375Q has a similar mutation to ADH1B2, so we hypothesized that this would also have an accelerated rate from ethanol to acetaldehyde. So this is a very simplified overview of the steps taken to prepare our enzyme for our functional assay. There are many steps that came before this to even begin, but first we introduced our enzyme into E. coli cells using a plasmid. We then extracted our enzyme from the E. coli and removed any cellular debris. Next, we passed our enzyme through a nickel affinity column where ADH4 binds selectively and other impurities are washed away. And then we're ready for our functional assay. To determine differences in enzymatic rates between wild type and R375Q, we wanted to look at this ethanol to acetaldehyde conversion. But while that's going on, there is a simultaneous process happening where NAD plus is being turned into NADH. NADH can be measured with a UV vis spectrometer at 340 nanometers. The higher the absorbance of NAD measured, the higher the amount of acetaldehyde that's also there. Results. So there are observable differences in enzymatic activity. Just the starting point alone is over 2 times higher in our wild type ADH4. And then you can see it has a positive slope. Our R375Q mutant showed no detectable activity. So what does this mean? So our wild type ADH4 showed normal conversion from alcohol to the toxic step. Our ADH4 R375Q did not produce any acetaldehyde. It had no detectable activity. This means the first conversion in our ethanol metabolic pathway is delayed. We could speculate this to mean an accumulation of ethanol in the blood. I'm curious if this could contribute to binge drinking because our toxic acetaldehyde is not being produced. So further research will be needed to better understand R375Q mutant. We would also like to collaborate with native biodata. They're an an institution that works with researchers to address issues pertaining to Indigenous communities. Indigenous genomic data, they house Indigenous genomic data, but are very selective with who they collaborate with. With everything I've learned, I wanted to reflect on my first statement. I began this research thinking my father was this horrible person. But now I understand that alcohol misuse isn't just an individual choice. There's other factors at play. There's this ideology that every decision we make impacts 7 generations before us and the 7 generations after us. Breaking cycles not only affects myself and my daughter, but it heals our family line, past, present, and future. It's a little unorthodox, but I'd like to end with my traditional introduction. Alyssia Begay Yanishya, Noda Edine Inishle, Tachini Bashichin, Tachini Dashache, Kisaana Dashanale. I am Alyssia Begay. My maternal grandmother is Ute Clan. My maternal grandfather is Red Runs Through River Clan. My paternal grandmother is Red Runs Through River Clan. And my paternal grandfather is Hopi Clan. Thank you.